Good evening, folks. I hope everybody is doing well tonight. This is part 3 in our ongoing lecture series on the genetics of Mendelian inheritance, looking at heredity and inheritance and the related concepts. If you have not yet watched part 1 and 2 in this lecture series, I recommend you go back and do that so that you have the necessary background to understand the more advanced concepts we're going to be getting into in this lesson. If you are in my biology class, you should definitely hop on the Blackboard site if you've not already done so and download both the handout on Mendelian genetics with a summary of important concepts and vocabulary, as well as the handout on the making of Punnett squares to help you use that particular analytical tool to predict genetic outcomes. By the way, if you watched the last lesson, like immediately after I stopped recording, my brain coughed up the data. It is Rudolf Virchow. Rudolf Virchow was the German geneticist who was, along with Erich von Schermach and Karl Correns, going to end up, Dutch geneticist, the other two were German, ended up independently rediscovering Gregor Mendel's research. So there's my last little detail that I left out at the end of last lesson. Okay, so last time we learned about how to analyze a crossing between two organisms if we know their genotypes, and to predict likely genotypes and phenotypes among the offspring. We did a monohybrid cross in which we're only studying one trait at a time. We did a dihybrid cross in which we're studying the transmission of two traits at a time. And we saw what it means to be a carrier and how you can be homozygous or heterozygous, and under what conditions you express a recessive trait and all that fun stuff. Now, the things we did last time, even the dihybrid cross, which involved that crazy 16-box Punnett square, all of those things were simple genetic cases. All of those were situations where we have a dominant paired with a recessive. It is not always that simple. In the world of genetics, you're not always going to have things that are nice, neat either-ors like that. And traits don't always fit into perfect little boxes that are discrete and easy to sort. Gregor Mendel, in his initial experiments, chose traits that all were like that because he was specifically looking for traits that had an unambiguous either-or for interpreting his results clearly so he could learn the pattern. But had he selected other traits, he might not have arrived at the pattern quite so easily, because we have learned since his time, and he even contributed some to this in the latter part of his research, that there are other ways this can happen, that there are other genetic phenomena that are a little more complicated. Let me begin by asking you a question. How tall are you? Okay, you can't answer me, but maybe you're answering. You can talk to the computer, I just won't hear you. But let's pretend I heard you say you were 5'6", or 5'3", or 5'11", or 6'2", whoa, you're tall, or 4'11", you're small, a whole range of possible heights, right? None of you probably, when I asked your height, had tall or short pop into your head. Because human height is not a binary, right? It's got a whole range of possibilities. There's a wide spectrum of possible heights. Even setting aside for a moment conditions like dwarfism and gigantism, the normal range of human height could go anywhere from like 4'6" for the shortest adults to more than 7 feet for the tallest adults. Obviously, those extremes are just that. They're extreme. You don't see those very often in individuals who aren't having some kind of glandular disorder or what have you. But that's still all within the range of normative human variation. But you're completely unsurprised, right, when you see somebody, male or female, who's like 5'7", 5'8", right? That sounds like a pretty average sort of height, doesn't it? Here's the thing. There's a whole spectrum of variation in there, a wide range of possibilities. We don't sort height into nice, neat either-or boxes like blue eyes or brown eyes would, for example. Height is an example of a genetic phenomenon called polygenism. Polygenism. Poly means many. Polygenism, many genes. What happens with a polygenic trait is that it is the result of not one gene, but a bunch of genes. That in polygenism, several genes all contribute to controlling the same visible character. In other words, there's not a single gene for height. There are a whole bunch of genes whose effects add up to determine what your height will be. And you should be able to see why that is, right? Obviously, the length of my skull is something. The thickness of my vertebrae are part of it. The thickness of my cartilage between the vertebrae are parts of it. The length of my thigh bones are parts of it. The length of my shin bones are part of it. the amount of growth hormone I'm producing is part of it. All of these are things that are going to contribute genetically to the total height that I end up having. There are more than 50 genes involved in contributing one way or another to this thing we call height. And none of them are controlling for height, they're controlling for shape, thickness, growth of particular bones or structures or whatever. But their additive results gives us the phenotypic character that we call height. Now, because you've got 50 or so genes all involved in that one thing, every individual one of those genes is going to follow the genetic rules all on its own. You're going to have some of them where there's a dominant and recessive pair. You're going to have some of them that are, fixed alleles where everybody's got exactly the same gene for them. You're going to have some of them maybe where there are two different dominant options and one recessive option. You're going to have some of them where the dominant option codes for being a little bit thicker or larger or taller. Some where the recessive option is the one that codes for being a little bit thicker or larger or taller. And your total height is the additive effect of all of these things. And that's why somebody can be taller or shorter for different reasons. You can be like, oh yeah, I'm shorter because I have little legs, or I'm taller because I have a long torso or whatever, because it depends on which genes are the ones responsible for that happening. The net result of this is that polygenic traits, rather than fitting into nice, neat either-or characters, have a spectrum of variation, a range of possibilities between the individuals at one end of the spectrum that have all the different genes that code for being taller, the very tallest people, and the people at the other end of the spectrum that have all the different genes for coding for being shorter. Most of us, for fairly obvious reasons, with 50 or more genes involved, are going to average out somewhere, right? We're going to come somewhere in the middle where we're going to have some of the genes that code for being taller and some of the genes that code for being shorter. And as a result, it's possible for us to say that a population has an average height, right? You're going to find that a lot of these traits will kind of vary on a bell curve, that at either extreme of the bell curve, you're going to have the very tallest people, the very shortest people, but you're going to curve up more towards the middle. The likeliest result is somewhere near the middle, which is why we can talk about a height of like 5'7" or thereabouts and say that's a pretty average height for a human, right? Now, depending on what trait you're looking at, the curve may not be a perfect bell curve. It might go one way or the other, and in a particular population, that curve might go one way or the other. For example, if you are a member of one of the tribes from the central Congo known as the Pygmies, their particular gene pool does not have a lot of the tall genes in it. They're mostly intermarrying with one another. Ergo, they're passing the short genes around generation after generation, and their average adult height ends up being a lot lower. So their curve leans, this is the middle, their curve leans sharply off to the short side. There are a lot of human traits that vary polygenically. Actually, eye color is a great one. Eye color is not a simple thing. Blue versus brown is simple. Brown is dominant, blue is recessive. But there are actually 5 different genes that all are involved in controlling eye color. And everybody genetically is either blue-eyed or brown-eyed. But then you start throwing those other genes in that affect hue or affect brightness or darkness of the pigments or affect the amount of the pigment produced or add other pigments to the mix. And it's from that you end up getting the hazel eyes and the green eyes and the various shades of brown eyes and the gray eyes and all that sort of thing. So yeah, although brown-blue is an easy dominant recessive, human eye color is in fact polygenic. Hair color, by the way. Hair color is actually a pretty straightforward one. It is technically polygenic, but there are only 2 genes involved. There's brown-blonde. Everybody genetically is either brown or blonde. Then there's a second gene, which you have or you don't have, that determines whether another pigment is mixed in, which would be the red tint. And depending on what your base color is, the red tint will give you either more of like a strawberry blonde, or more of like an auburn kind of color. Totally separate, unrelated genes control the amount of pigmentation that you produce, which can cause your brown to shade into black or your blonde to be a more like dirty blonde or a more pale blonde or what have you, but that's affecting amount of pigmentation, not the actual color. So yeah, you can say hair color is technically a polygenic trait as well. Brown blonde is easy, but then there are other things that play into it. So we actually think right now in modern genetics that more traits are polygenic than not, that a lot of the things that happen in our bodies are the result of more than one gene. And this can also account for why certain genetic disorders are worse in certain people or better in certain people or respond better to one treatment or another, because we think that some of these disorders, which we diagnose on the basis of symptoms, may be the result of different combinations of genes. And depending on which gene combination you have, one treatment might or another might or might not be effective. Now, I have to point out, and this is a really important one for cultural reasons more so than for scientific reasons, is that one of the traits that varies polygenically in human is our skin color. Pigmentation is a polygenic trait. There are dozens of genes that affect the color, tint, and amount of pigment that your body produces, as well as its distribution. As a result, sorting humans into groups on the basis of skin color is frankly kind of stupid, because there are dozens of genes that affect your skin color, at least 14 primary ones and like another 30 or so that have something to do with it. And as a consequence of this, the combination of genes you have for pigmentation may not even be the same combination of genes as somebody who's basically the same color as you, because there's more than one way to arrive at the same mix. And none of those genes are localized to any one specific population in the world. You'll find that most of them have to do with how close to the equator that population came from. Closer to the equator you get, the darker pigmentation tends to be, because it's good defense against intense solar radiation. That's what pigment is for. It's there to soak up UV rays, which is why you tend to see darker people in equatorial Africa and paler people in Scandinavia. So yeah, you can plot human skin color on a map of the globe, and you'll find the further north you get, the paler people tend to be. There are obvious exceptions. Migration can change that around. A population can move from point A to point B and take their skin pigmentation genes with them. And that's the other reason why sorting people into racial groups on the basis of skin color is stupid, because humans move around and they mix with other humans and they intermarry and they pass their genes all over the place. There is no suite of genes, no specific cluster of genes or even specific gene I can look at that says, okay, this is the gene for being black or white or whatever, because there isn't a gene for those things, because biologically those aren't things. Those are labels that we attach to a certain category of phenotypic traits. Now, culturally, as a cultural phenomenon, yes, race is a real thing. It's a cultural and social construct. But please understand, it is not in any way a biological construct. It's something that we attach as a label to particular suites of traits based on visible identifiers and cultural features that we attach to them. Now, because certain groups of people tend to live in certain regions, and because, here's a fun statistic for you, 90% of people will have children with somebody from within 50 miles of where they were born, even if they move further away. And as a result, clusters of genes tend to form in local areas. People pass the same genes around again and again within their little local gene pool. And as a result, folks who live in a particular region tend to resemble one another a bit more over time. And you'll tend to say, okay, yeah, that's why we have lots of pale people in Scandinavia, and that's why we tend to have lots of darker people in the Congo, and so forth, because the same genes are being passed around in that local zone. However, people leave those zones and mate elsewhere. People come into those zones and mate within them, and they bring their unique genes to the party. So yeah, human genomes are way too mixed up, and there's too much variation to allow skin coloration to be a meaningful identifier of this thing that we call race. Race is it is absolutely a cultural construct. It is absolutely not a biological construct. And as I like to point this out, you're looking at me giving this lecture right now. You probably are thinking I'm a white guy. Nope. Culturally, I am not. I'm just really pale as my people go. All right, so, moving on from that, though, you might think to yourself, okay, so why is it then that African Americans are more prone to sickle cell anemia? Or why is it that Ashkenazi Jews are more prone to Tay-Sachs syndrome and things like that? The reason for that, folks, is because of that precise phenomenon I mentioned before, breeding within your own local gene pool. It means the same genes get passed around again and again and again. And that can mean within a geographic zone, but it can also mean within a cultural zone. Keep in mind that the overwhelming majority of African Americans came to the United States as a result of slavery between the 1600s and the 1800s. That most African Americans have slave ancestors that were involuntarily brought to the United States, and that gene pool has largely been an African American gene pool ever since, because for cultural reasons having to do a lot with segregation and with racial discrimination and all sorts of things, African Americans were way more likely to mate with other African Americans than they were with people of other ethnic groups. And as a consequence of this, the African American gene pool was a lot more closed off than from the rest of the population. Ergo, the same genes got passed around in that gene pool again and again and again for centuries. And it just so happens that a lot of African Americans were all taken from, well, a lot of the slaves, I should say, were all taken from Africa, from a little region of Africa called the Slave Coast. Well, that's what they called it back in the day. These days it's around Liberia, Sierra Leone, Côte d'Ivoire, that part of Africa, like the western region of Africa. But that was the major site for capturing slaves to bring to the New World. And as a result, the gene pool of that little section of Africa, where it happens that the sickle cell gene is very, very common, is disproportionately represented in the African American population. And because that population has been interbreeding within itself for centuries, that means that that gene got passed around a lot and became kind of concentrated there, you might say. So statistically speaking, an African American is way more likely to carry the gene for sickle cell anemia than a non-African American here in the United States, or indeed more likely than a native African at this point. So those kind of phenomena are the reason why you get certain disorders that are common in certain populations. Likewise, if you convert to Ashkenazi Judaism and you have no Ashkenazi Jewish ancestors, it does not make your kids more likely to have Tay-Sachs syndrome. Tay-Sachs syndrome is a rare and rather terrible recessive genetic disorder. It basically causes the central nervous system to break down. You will almost certainly be dead by the age of 5 if you have it, so you don't survive to adulthood in any case. But Tay-Sachs is only 1 in every 400,000 or so live births in the population at large, but 1 in every 4,000 births among Ashkenazi Jews, Jews of Eastern European descent. And that's because cultural traditions among Ashkenazi Jews, as well as marginalization within Eastern European society, meant that Ashkenazi Jews almost always married other Ashkenazi Jews. And at some point in the distant past, that gene crept into the Ashkenazi gene pool and basically stayed there and got passed around within it ever since, which is why if you're of Ashkenazi heritage, you are way more likely to carry the gene for Tay-Sachs syndrome than a random person on the street is. So that's why you end up with these genes that we associate with certain ethnic groups. Do not fall into the trap of making you think that means that race is a biological feature that can be identified by those traits, because it's not uniform in any way and it's not consistent in any way. You can get the same skin pigmentation in 2 entirely different ways in 2 entirely different regions of the world. You bring in an Aboriginal tribesman from the heart of Australia, they'll have the same skin coloration as a Congolese tribesman from Africa, even though those populations have no recent common ancestor from which they got that common pigmentation. And they get it by 2 different genetic combinations. All right, sorry, I lost my train of thought completely. It went totally off the track. Okay, just, all right. But one last thing. Yeah, okay, I got the train back on the track. So y'all do these tests nowadays. I went and did it too, the Ancestry.com genetics test, or 23andMe, or National Geographic, or any other companies that you can spit in a little tube and they'll send you back genetic information about how much Neanderthal blood you have and so forth. And a lot of these are going to come back and say, okay, well, you're 12% Swedish or 15% Northern African or what have you. There's no gene for being Swedish. What they're saying basically is, okay, in certain regions of the world, because of that inbreeding phenomenon that I told you about, where people tend to breed with other people who live near them. If you don't know why you need to breed with people who live near you, I can talk to you quietly in a private conversation. But in any case, though, because of that phenomenon, because people tend to mate locally and the same genes get passed around in the same gene pools over and over again, there are certain genetic variations that are more common in certain regions than in other regions. And a lot of them aren't even coding for any visible, obvious trait that we would see in you. A lot of them are even intron sequences that are just being passed on within populations that don't code for anything at all. They're just kind of there. And the ones that are actually introns are more likely to stick around in a population because natural selection is not acting on them, because they're not good or bad for you, and therefore they're just carried along for the ride with every other gene that the person has. So what we're really testing for in those kind of tests is to say, okay, like 75% of the people globally who have this genetic variant right here have ancestors from Sweden. So if I see that represented in your chromosomes to this extent, we can calculate that you're likely to have, based on this variant, and that variant, that are all well represented in Swedish populations, that you are likely to have a Swedish great-grandparent. So we'll say you're 12% Swedish or whatever. Again, it's not a perfect thing. Those results can surprise you at times, and some of them are really strange, and some of them spit out results you might not have expected, and sometimes you find out the dad wasn't actually dad or whatever, but that's the fun that comes with this. So yeah, there's not a gene for being Swedish or Northern African or Brazilian or whatever, but there are genetic markers, if you will, that are more common in certain parts of the world than others, and therefore more likely to be represented in certain populations than in other populations. And the bigger a database we build of people reporting their own genetics and reporting their own ancestry and so forth, that's why it's really cool to have genealogy websites like Ancestry doing this, because they can match that kind of stuff up. We're learning more and more about what gene markers came from what gene regions of the world, which means these tests are getting better over time. But I don't want you to make that thing that validates the idea of race as a biological construct. It does not. If anything, it validates the idea that human variation is extremely broad and extremely mixed, and trying to sort people into racial groups on the basis of anything biological is frankly not only wrong, but dangerous. Because it ends up, every single time we've tried to do it historically, it has ended up leading to all kinds of racism, eugenics, Jim Crow laws, white superiority, all kinds of bullshit like that frankly we don't need. All right, so polygenism, though. Polygenism is telling you what happens when you got several genes all affecting one trait: skin color, average adult heights, body fat percentage that you store. Incidentally, that is another one that is genetically controlled. If you're dieting and you hit a plateau and you can't get any lighter, you've hit your natural plateau. Don't try to push beyond it. You're going to hurt yourself. So there are lots and lots of things in our body that more than one gene affects them. Probably more things like that than not. So polygenism is pretty common. But let's flip it around. Let's go the other way. Rather than having one thing all affected by several genes, let's talk about what happens when you have several things affected by one gene. That is a phenomenon called pleiotropy. P-L-E-I-O-T-R-O-P-Y. Pleiotropy. Please be careful with how you say that. Somebody always wants to say pleiotrophy on my test and put an H in there. There's no H in there. Pleiotropy. So pleiotropy is the phenomenon you get when you have a single gene that controls multiple characters, one gene that does several things. This can happen for a couple of reasons. This can happen because the gene is making a protein that is used in several different parts of the body, and therefore that is affecting a bunch of different things about you. It can also happen because that one gene can be spliced more than one way to make more than one different protein product whenever you translate it. If you haven't watched my lesson on RNA yet, go back and watch it and learn about splicing. But suffice to say, we've learned that it's not as simple as one gene is the code for one protein. It's a lot more complex than that. And sometimes you can take a single gene, cut it up and put it back together in different ways and get more than one protein product out of that same stretch of genetic code. Ergo, a single gene can, in fact, code for different things in your body that may not be related to each other in any way. So that's what happens in pleiotropy. You have a single gene that does multiple different things in your body, and therefore having different variants of that gene will control more than one part of you at once, and they'll cause change in more than one part of you at once. Now, the coolest thing about pleiotropy, and here's the thing that tends to break people's heads, so do your best to follow along with me here, is that pleiotropy is a situation where one gene does more than one thing. It is possible with a pleiotropic gene for some of the things it does to be dominant and other things it does to be recessive. In other words, we can't look at the gene and say, oh, that's a dominant gene or a recessive gene. We could look at it and say, oh, this gene behaves as a dominant for this thing that it does, but it behaves as a recessive for that thing that it does. That the gene can be viewed as either dominant or recessive depending on what effects it's got. And so what this means in practical terms is that this does something different than the Mendelian genetics we talked about last time. I've kept saying to you, okay, it doesn't matter if you're homozygous dominant or if you're heterozygous, right? Because either way, you express the dominant gene, and it's going to look exactly the same. Mendel's homozygous dominant purple flowers looked exactly the same as his heterozygous purple flowers that were carriers of a white gene. Doesn't matter. Dominant gene gets expressed either way. But in pleiotropy, it matters. Because in pleiotropy, it matters how many copies of the gene you've got, because if you have one copy of a pleiotropic gene, you only get the dominant effects from it. Because if that gene has a recessive thing that it does, you have to be homozygous for a recessive gene in order to express its effects, right? So if you have one copy of a pleiotropic gene, you will express any dominant effect of that gene. If you have two copies of that pleiotropic gene, in other words, if you are homozygous for it, you will express both the dominant and the recessive effects. So you'll get more things out of the pleiotropic gene if you're homozygous for it than you will if you're heterozygous for it. You'll get more of its effects coming out. So it actually matters in pleiotropy whether you are homozygous dominant. Sorry, I accidentally paused myself there. So it really matters in pleiotropy whether you're homozygous dominant versus heterozygous, because heterozygotes only get the dominant effects of the gene. Homozygotes get both dominant and recessive effects of the gene. Let me illustrate how this works. Let's use a well-known example. If any of you are fans of the TV show Game of Thrones, you might know an actor by the name of Peter Dinklage. Peter Dinklage plays the character Tyrion Lannister, and it's an important plot point in the series that Tyrion Lannister is a dwarf, that Tyrion Lannister has dwarfism, and specifically the actor playing him, Peter Dinklage, has a form of dwarfism known as achondroplasia. Achondroplasia. Achondroplasia is a connective tissue disorder caused by a particular variant in the gene for the formation of our connective tissue, particularly the collagenous fibers within our body, that, as you can see, causes multiple effects in the individuals who have it. It's the most common form of dwarfism. There are like 11 different forms of dwarfism, but in this particular form of dwarfism, it has characteristic, certain body shape that goes with it, relatively short limbs compared to the torso, relatively large head, and short neck compared to the torso. You don't actually see it, but it also comes with certain characteristic heart defects that are relatively common in achondroplasic individuals that are less common in the population at large. Now, if Peter Dinklage is a guest star in my video right now. Peter, you want to come over? No? You're playing Animal Crossing right now? That's cool. That's cool. Yeah, I don't mind. Go take care of your island. All right, he's busy. So, anyhow, if Peter Dinklage were to show up right now, I would be able to look at him and know for a fact what his genotype is. I would be able to look at him and say that his genotype was that one right there. That he's heterozygous. Now, you see a Punnett square here involving a big D and a little d, right? And you might ask yourself, well, how do I know that Peter Dinklage is heterozygous? Well, here's the thing. The condition of achondroplasia is a dominant condition, which just goes to show a really important thing, that dominant does not always equal, the positive or common trait in genetics. That dominant can be a rare trait, dominant can be a disorder. Huntington's disease, which is a very lethal genetic disorder, is a dominant condition. Achondroplasia is a dominant gene. If you do not have achondroplasia, your genotype for that particular locus is the bottom right. You are homozygous recessive. You have two little d's, which also just goes to show that recessive does not automatically equal rare or bad. So that's the most common genotype for people. Most of us have the two little d's. Most of us are homozygous recessive at that locus. So if Peter comes over, well, I know he's heterozygous. Achondroplasia is a dominant condition. But you might be thinking to yourself, well, how do we know he's not homozygous dominant? How do we know he didn't get the dwarfism gene twice? The way we know that is because he's alive. Because there is a recessive effect attached to that same gene. If you get one copy of that gene, you get the dominant effect only, which is the condition we call achondroplasia. If you get two copies of that gene, you're homozygous for it, you get both the dominant and the recessive effects. And it happens that the recessive effect of the gene for dwarfism is a lethal defect that, if it does not kill the child before birth, will cause them to die within a couple of days of birth. In other words, you can't survive if you are homozygous dominant for that gene. And the fact that Peter Dinklage is alive and walking around means that, in fact, he has that gene. One copy, not two. He has to be heterozygous. Now that leads to an interesting situation. And this Punnett square right here shows you what happens if you have a monohybrid cross between two individuals that both have the gene for achondroplasia. Both of them have one dominant gene for achondroplasia. Both of them have one recessive gene for normal collagen formation. This couple, this dwarf couple, they're both be dwarves in this instance, has a 1 in 4 chance of conceiving a child that inherits both dominant genes. You see I put a little tiny X in the D there to indicate the hidden recessive in there. That's a couple. The child will certainly have dwarfism, but the child will also have the lethal defect and will die at or shortly after birth. This couple has a 2 in 4 chance of having dwarf children who got one copy of the dominant gene for achondroplasia and one copy of the recessive gene for normal collagen formation. And then we have a 1 in 4 chance that this dwarf couple will have a homozygous recessive child that is not going to have achondroplasia at all. That child will have normal growth and development, and that child is not a carrier of anything because you can't be a carrier of a dominant gene. And therefore, that child has no more chance than anybody else of having dwarf children. So that's how these things tend to go with pleiotropic genes. If they've got a dominant and a recessive effect, dominant effect comes out with one copy, dominant and recessive effects come out with two. Let's go back to a genetic disorder that I said I was going to use as an example again. Let's talk about sickle cell anemia for a moment. Now, the gene that causes sickle cell anemia is pleiotropic. It has both dominant and recessive effects. The disease that we call sickle cell anemia is the recessive effect of that gene. In other words, for you to have sickle cell anemia, you've got to have two copies of it. You have to inherit it from both of your parents. If you've ever had a blood test for this and the doctor told you you have quote-unquote sickle cell traits, that's the common usage in medicine. That means you have one copy of the sickle cell gene. In other words, that you are not expressing it yourself. You do not have sickle cell anemia, but you've got the gene for it present in your DNA, which means your kids could have it. You're a carrier of that gene. In other words, you're heterozygous. You have one copy of the dominant gene for normal hemoglobin formation, one copy of the recessive allele for sickle cell anemia. And by the way, that is kind of sloppy usage to say sickle cell trait does not match up with the way we actually talk about things in genetics, but it came into medicine at some point, and that's how people tend to describe it. So that's what that means if somebody tells you you have sickle cell trait. You're a carrier of the gene for sickle cell anemia. So for you to have sickle cell anemia, you have to be born with two copies of the gene. It's a recessive condition. It will not be expressed without that. In other words, both of your parents had to either have it or had to be carriers of it for you to end up with it. So you end up with two copies. That's unfortunate. You got sickle cell anemia. Let's suppose you end up with one copy of that gene. You're a carrier for sickle cell anemia because that's the recessive effect. It just so happens that the gene for sickle cell anemia, being pleiotropic, has a dominant effect and a recessive effect. If you get one copy of the gene, if you're heterozygous and you have one copy of normal hemoglobin gene as well, you don't have sickle cell anemia because that's recessive, but you do get the dominant effect of the gene. If you get two copies of it, you get both the dominant effect and the recessive effect, which is sickle cell anemia. You know what the dominant effect of that one is? The dominant effect of that gene is resistance to malaria. Malaria is not a disease we have to worry about here in the United States, but in a lot of tropical areas, malaria is a serious problem. It still kills 500,000 people around the world every single year, and it's something we should really be working harder to eradicate. There are efforts to do so, but it's been slow going because, frankly, there's not been nearly enough investment in it. But here's the thing. Malaria is a disease that is a serious problem in Western Africa. Thousands of people still die of malaria in Western Africa every single year. If you happen to live in Western Africa, having one copy of the sickle cell gene is a really good thing. You won't get sickle cell anemia out of it, but you'll get resistance to a very dangerous disease in your environment. Now, remember how I said that African Americans are disproportionately taken from regions of Western Africa as slaves, or they were back in the day? That means that that gene pool in which the sickle cell gene was common was disproportionately sampled in bringing slaves to the New World. And therefore, most modern African Americans, being descended from that population, are more likely to have inherited the sickle cell gene than Americans from other parts of the world ancestry. Therefore, we tend to think of sickle cell anemia as an African American disorder in the United States. It doesn't say you can't have it if you're not African American, but you're way more likely to have the gene for it if you have African American ancestry, specifically because that gene came from a region of Africa in which having one copy of it was actually beneficial. Now, sure, that means the occasional child was born over in that region with 2 copies of the gene, and that's unfortunate, but they still are resistant to malaria. So, ends up natural selection favors the persistence of that gene because it defends you from a seriously lethal disorder. All right? Okay, so that's pleiotropy. So we now dealt with what happens if you have one gene affecting several different things. We've dealt out with what happens if you have several genes all affecting one thing. Let's look at a different phenomenon. What if there is no dominant gene at all? It's not a requirement. It's possible that, remember, dominant versus recessive is a chemical property of the structure of the gene. If a gene is dominant in me, it's dominant in you. If I am born with 2 copies of a dominant gene, if I'm homozygous recessive for it, it's going to stay dominant in all my kids. If I am born with 2 copies of a recessive gene, I will express it. That did not make it become dominant. It's still a recessive gene, and when I pass it to my kids, they'll only express it if they also get it from the other parent, and therefore, they're homozygous recessive too. So it's conceivable that the most common gene in the population can be a recessive, as we saw with achondroplasia. The gene for normal cartilage formation is a recessive condition. Now, how about this, though? Let's suppose we have 2 different alleles available at a locus. There are 2 gene variants in a population for a given trait. There's no guarantee that one of them has to be dominant. Maybe both of them are recessive. Maybe neither of them is dominant over the other. If that's the case, we're going to get a phenomenon known as incomplete dominance. Some older textbooks called it partial dominance. It's D-O-M-I-N. It's a D-O-M-I-N-A-N-C-E, dominance. Incomplete or partial dominance. So in incomplete dominance, there is no dominant gene presence. Here's what happens. There's an ornamental flower, you can look them up if you want online, called the Japanese 4-o'clock. Japanese 4-o'clocks have 2 different genes for color. There's one gene for red Japanese 4-o'clocks and one gene for white Japanese 4-o'clocks. But you know what? Neither of those genes is dominant. So if I cross a red Japanese 4-o'clock with a white Japanese 4-o'clock, I get a pink one. Now, you might be thinking to yourself, well, Mr. Davis, didn't you say that blending isn't a thing? That genes do not blend or, combine into one new gene? They didn't. What happens in a case of incomplete dominance is that each of the 2 genes has a partial effect. Each one is partly expressed. The consequence of that is a blended phenotype. The phenotype is intermediate between the 2 genes. But genotypically, they've remained separate and discrete as Mendel's laws said that they should. So in a case of incomplete dominance, you will have what appears to be blending. You'll have an intermediate phenotype. But we can prove that it's not by doing a test cross in which we, actually not a test cross, in which we, let's prove that it's not. The way we prove that it's not is we take the blended phenotype and we let it breed with itself, or let it breed with another one with the same blended phenotype. Here's the Punnett square for that. You can see that we have color, C superscript R for red, C superscript W for white. Now, I have told you that these have not blended together. They've remained separate and distinct. If they had blended into one gene, if they had combined into one single pink gene, I would expect that when this thing self-pollinated, that all the offspring would be pink. But look what happens here. Each one has a 1 in 4 chance of passing on each of its 2 genes, because the red and the white did not, in fact, blend together. They remain separate and discrete within the individual. So this plant has a 1 in 2 chance of passing on its red or its white. If it's self-pollinating, it's crossed with itself, has a chance of passing on red or white. That means that a pink Japanese 4-o'clock crossed with another pink Japanese 4-o'clock has a 1 in 4 chance of getting red offspring because the red gene was passed on by both, a 1 in 4 chance of getting white offspring because the white was passed on by both, or a 2 in 4 chance of getting more pink offspring where 1 red and 1 white showed up. In other words, the fact that we see segregation of the alleles in the F1 generation, or the F2 generation here, proves to me that the genes did remain separate. If they don't fuse into one pink gene, I just see pink forevermore. But the fact that red and white come back out shows me that incomplete dominance was at work. Human example of this, let's talk about hair texture. There are two genes for human hair texture, straight and curly. You got one or the other, unless you got both. If you got one gene for straight hair and one gene for curly hair, you end up with what we call wavy hair, because neither of those is a dominant gene, and you got a texture that's in between, not fully straight, not fully curly. Now, I know what you're thinking. You're thinking to yourself, well, hey, my hair is straight whenever it's humid out and curly whenever it's not, or something like that, or, you know, after I wash it, it relaxes more and gets straight, or whatever. That's because this is a great example of the fact that your phenotype is not strictly determined by your genetics. Your genetics say what the baseline conditions are going to be, but the actual phenotype you end up expressing has a lot to do with the environment that you live in. There are lots of outside factors that can either affect the expression of a gene or change a feature of the body post-expression. Like hair color and texture are a great example. Keep in mind that your hair, the part you see above the scalp, all that hair is dead. That's a dead shaft of skin cells. The only living part of the hair is deep down inside the root. All the parts that are above the surface are just dead cells, which means they can't repair, they can't regulate, and that means they're really subject to environmental damage. And let's be honest, we put a lot of crap in our hair, don't we? Shampoos and conditioners and hairsprays and products of different sorts. We put all kinds of chemicals in our hair which can soak into the hair and modify either its color or its texture. Furthermore, you all know you spend a lot of time out in the sun, your hair tends to get a little more bleached, right? Or if you spend a lot of time swimming in a swimming pool, your hair tends to get a bit bleached. There are lots of things we can do to our hair that cause its color to change. It does not change the genetics of your hair one bit. Furthermore, as you get older, your hair tends to change color, right? Genetically, it's still the same color it always was. What's happening is that pigment-producing cells associated with your hair follicles are gradually shutting down over time. And that means that when they're not producing your pigment, the hair becomes more of a gray or a white color, depending on what its base color and texture were. Thicker hair looks more gray, thinner hair looks more white. But yeah, as you stop making pigment, the hair becomes unpigmented. That's what ends up happening. And genetically speaking, you're still exactly the same hair color you always were. So that's a case in which a developmental feature, you know, the shutting down of pigment cells over time, affects the phenotype that you have. Even when you've gone completely white in old age, you're still genetically brown-haired, or whatever hair color you happen to be. Suppose I have lots of genes that code for being taller, but I was malnourished as a kid. You know, my parents locked me in the basement and, you know, threw down meat every 2 days, you know, and that didn't actually happen. But, you know, suppose you didn't get proper nutrition as a child, you might not have grown to your full genetic potential, and by the time you finished your growing years and your growth hormone declined, you weren't as tall as the DNA said you should be. So genetically, I could be 6 feet tall, but if I was malnourished as a kid, I might only get to 5'4", 5'5", right? So there are lots of factors like this that can affect your phenotype. So I want you all to keep in mind that what you see is not necessarily what you get, that the visible phenotype you express is an interplay of genetic features and environmental features. So it's conceivable that even some genes that are in your DNA are not being fully expressed in all the ways that they can be at all times. Think about how many changes go through your body at puberty. That means you had lots of genes the whole time that were not being expressed. A lot of people, for example, their hair color gets darker at puberty. If you'd seen me when I was like 8 years old, I had white blonde hair. And then when I hit puberty, it all started changing the color you see right now. Well, a little less gray than you see right now, but color in that vein, because that's not at all an uncommon thing. Skin color and hair color both tend to darken a little bit with puberty because of the production of more pigment-producing cells. So just keep that in mind, you know, that you can't always look at phenotype and get an accurate assessment of genotype, and hair in particular is subject to a whole lot of environmental forces that can change up the apparent color that it has. So that's incomplete dominance. That's what happens when there's no dominant gene. Let's try it a different way. What if there's more than one dominant gene? Let's suppose you're heterozygous and you've inherited 2 different genes and both of them are dominant. In that case, you're going to get a phenomenon called codominance, C-O-D-O-M-I-N-A-N-C-E, codominance. In incomplete dominance, neither gene was dominant, so each one had a partial effect. In codominance, both genes are dominant, and therefore both genes are fully expressed. You express all of both genes. You have more than one trait for that character. Codominance appears in a lot of animal fur patterns, a lot of snakes. If you look at a lot of snakes, if you know anybody who breeds snakes, you can ask them about this, that a lot like diamondback rattlesnakes, that certain diamond colors are dominant while certain base colors are also dominant. You can breed together two snakes and get a mixed pigmentation that's really pretty from doing that because both colors are dominant, and therefore they'll both appear in the snake's scales when you breed them together. By the way, just to dispel a common myth about this, if you see somebody with one brown eye and one blue eye, that is not codominance. That is actually a recessive genetic phenomenon called heterochromia iridium, Heterochromia, heterochromia iridium, IRIDIUM. Heterochromia iridium is a bilateral condition which causes one half of your body to make less pigment than the other half of your body. Ergo, one eye will be darker than the other. And that can often manifest as one eye being brown and one eye being like blue or like a pale green or whatever, or gray or whatever. If you know somebody with that condition with one brown eye and one blue eye, and if you know them really, really well and they're willing to strip down for you, you will see that one whole half of their body, in general, tends to be paler pigmented than the other. Okay, so that's codominance. You have two different traits for the character. Human example of codominance. Yeah, because heterochromia iridium is not codominance, it's a recessive disorder. But human example of codominance, and one of great medical relevance, and I know a lot of you listening to this lesson are going into medical fields, and all of you have blood, so this will be relevant to you, is your blood type. Now, you all hopefully know your blood type at this point in time, A positive or B negative or whatever it happens to be. Now, if you don't know that, go donate a pint of blood. We need blood right now. Donor banks are desperate. You save somebody's life, and they'll tell you your blood type if you don't already know it. But anyhow, human blood type, by the way we report it, we list it as a letter and a positive or negative. Those are two completely unrelated things. Those are two different features of your blood that we group together for medical convenience. The positive-negative part of your blood type is a dominant versus recessive that follows all the normal Mendelian rules. Positive is dominant, negative is recessive. So if you get two copies of the positive gene, your blood type will be positive. If you get one positive, one negative, your blood type will also be positive because positive is dominant and negative is recessive. If you get two copies of the negative gene, your blood type is negative. Positive is far and away the more common. 85% of humans worldwide have type positive blood. There's nothing bad about being type negative despite the name. Basically, type positive means that you have a certain antigen present on your red blood cells called D antigen. I don't know why they called it D as opposed to anything else. Type negative means you don't have D antigen. So set that aside for now. Let's not think about the positive negative part of your blood type for now because it has nothing to do with codominance. It's a straight up normal dominant versus recessive. Let's talk about the letter group of your blood type, the ABO group as we call it. There are three possible genes for that: A, B, and O. These genes code for specific antigens, cell surface markers found on the surface of your red blood cells. Type A is one of those antigens. We typically write it in genetics as capital A superscripted, capital I superscripted A. A is dominance. Type B is a different antigen. We write it as capital I superscript B. It is also dominant. Type O is recessive. That is the condition of not having a letter group antigen on your red blood cells at all. And type O is the recessive condition, so we represent it with a lowercase I. So capital I superscript A is type A antigen. Capital I superscript B is type B antigen. Little I is recessive. That is type O, which is the absence of an antigen. And yet another good example, by the way, of the phenomenon that tells us that recessive is not automatically rare. About 62% of humans worldwide are type O blood, even though it's the recessive condition. Okay, now what does this determine about? There's a common belief in the Far East that your blood type determines your personality. It's kind of an astrology sort of thing. If you're reading somebody's personal profile in China or Japan, they might say, oh, I'm type A negative or B positive, you know, in the same way that here in the United States, your profile might say I'm a Scorpio or I'm a Pisces or whatever. No truth to that whatsoever. There's really no clinical relevance to which blood type you have. It's basically, do I have this kind of marker on my red blood? Well, there is clinical relevance, but there's no actual health effect on you, which blood type you have. It's like, okay, this marker is type A. I have that particular marker on my red blood cells. Type B, I have that marker on my red blood cells. Type O, I don't have a marker for that at all. Here's what's up with that. A is dominance. There are two ways, therefore, to have type A blood. Wait. There we go. Sorry. Sit down. Okay. There are two ways, as you can see here, to have type A blood. You can have either two copies of the A allele. You can be homozygous dominant for it. It's a dominant condition. Or you can have one copy of dominant A and one copy of recessive O. A is dominant. You will express the A allele. There are two ways to be type B. You can have two copies of the B allele, homozygous dominance, or you can have one copy of dominant B, one copy of recessive O. Dominant gets expressed. You're a carrier of type O, but your blood type is B. There's only one way to be type O. O is recessive. Therefore, you have to be homozygous for it. Get it from both parents. But there's one other possibility here, isn't there? That other possibility is maybe you get type A from one parent, you get type B from the other parent. They're both dominants. Therefore, you will have both the A marker and the B marker present on your red blood cells. Both genes are fully expressed because both of them are dominant. You don't get a blended marker that's got the intermediate between the two. Your red blood cells literally have both A markers and B markers on them. Ergo, you are type AB. AB is not a separate blood type, folks. We list it as one for medical purposes, but in reality, if you're type AB, what you really are is type A and type B at the same time. You are both of those blood types simultaneously. Now, when type AB folks reproduce, that means they pass on either of those two. They will pass on one of those two, either the A or the B. In other words, type AB folks can't have type AB kids unless they pass on one and their partner happens to pass on the other. In other words, if you're type AB and you end up with a type AB kid, it's a coincidence. It's not because you were AB, because you only passed on either your A or your B. AB is not one blood type. AB is codominance, having two blood types at the same time. Now, here's a fun little phenomenon. Let's suppose, I drew this square to represent exactly what happens here. Here is a couple in which one parent is type A with a recessive O, one parent is type B with recessive O. This couple has a 1 in 4 chance of having an AB child. A came from one parent, B came from the other. They have a 1 in 4 chance of passing on the type A from one parent and the type O from the other and having a type A child. They have a 1 in 4 chance of passing on the B from one parent and the O from the other, having a type B child. Or they have a 1 in 4 chance of passing on both of their type O genes, ending up with a homozygous recessive type O child. In other words, this couple right here can have a child of any of the 4 types. It's the only way that can happen. Now, if they have that AB kid, and that AB kid partners up with a type O parent, draw the Punnett square for yourself. Go ahead and do that right now. AB parents, O parents. Just going to give you a minute to do that. I'm going to check my phone while you do that. Is anybody messaging me? Weird news in the world. All kinds of stuff going on. Okay. All right, so anyhow, though, if you drew that Punnett square, what you should have seen is that every child got an O from their type O parent, because you had to be homozygous to be type O, and then the AB parent passed on either their A or their B. In other words, that couple, the AB couple and AB parent and O parent, has a 50-50 chance of having a type A child or a type B child. There's no possible way for them to get a type O child or a type AB child. All right, and you can play with this as well if you want to play with blood type genetics. I didn't draw out all the possible Punnett squares, but it's easy to do. I showed you all the possible combinations there. You can draw combinations for anything that you like, and if you want to figure out your own, you can say, okay, here's my blood type, here are my parents' blood types. You can probably work out a Punnett square that would show how you got that way. By the way, clinical relevance of this, and a little bit of digression here from our genetics, is that in clinical terms, the reason why blood type matters is because all of these things are talking about cell surface markers, which you might recall your body uses to identify whether a cell belongs or does not belong. And what that means is that if you are given a pint of blood, if like you have a blood donation, and it has the wrong marker on it, your body will reject it. Your body will say, uh-oh, I don't know what that is, and it will think that the red blood cells are a foreign invader. So if you get a blood of the wrong type, you will have a transfusion reaction, as we call it. Your body will rapidly destroy the red blood cells. It will release a flood of compounds from inside those red blood cells into your bloodstream all at once. It will basically choke your kidneys, and you will die of kidney failure in about a week. Only way to save you if you get that is to have a complete blood transfusion, in which we take all the blood out of your body and put in new blood before your kidneys fail. All right, but so clearly that's a bad thing, right? And in the early days of transfusion medicine, we didn't know about that because we didn't discover blood types at first. When the first blood transfusions were done in the late 19th century, all they knew was, okay, sometimes when people have a blood transfusion, they die a week later and no one knows why. They learned pretty quickly, okay, if you get the blood from a relative, you're less likely to die a week later, but we still don't know why. But in the early 1900s, we figured out blood groups, and then we realized, okay, here's why sometimes you die from a transfusion. We figured out the different blood types, and therefore we're able to start banking blood safely and giving it to people according to their blood type. What's a safe blood type for you? Here's the rule. You cannot safely receive any antigen your body doesn't make. In other words, you can only get blood with antigens that you recognize or without antigens. So your body will reject any blood with an antigen that it does not already know how to make. So I am type B. That's my personal blood type. I am B positive. My body knows the type B antigen. So you can give me type B blood and I'll be just fine. If you give me type A blood, my body does not recognize the A antigen. I will rapidly hemolyze those red blood cells, and I will die of transfusion reaction in about a week. Can I safely get type O blood? Yep, I can safely get type O because O, remember, is the absence of a letter group antigen. In other words, there's nothing on a type O red blood cell to set me off. There's no letter group to react against. Ergo, my body will say, okay, that's cool, and it will not respond to the type O blood. So as a type B person, I can also safely receive type O. Can I get AB? Hopefully you said no. Because even though my body knows the B antigen on those cells, it does not recognize the A antigen. Remember, the A and the B are both present on my AB cells. Ergo, even though the B is fine, the A is not. Ergo, my body will reject the AB cells. So as a type B person, I can safely get type B or type O. My body will reject both A and AB because of the A antigen. Likewise, if I am type A, my body's cool with type A blood. It's also cool with type O blood because it doesn't have an antigen to set me off. My body will reject type B or type AB because of the B antigen. If I'm type O, only blood type I can get is type O because my body does not recognize either A or B, and therefore any other blood type will set off a transfusion reaction. If I'm type AB, I can get all of the above because my body knows both A and B antigens, so both A and B blood are cool. AB blood is obviously cool, and type O still doesn't have anything that'll set me off, so AB can get any of them. By the way, with the positive-negative part of your blood, positive means you have D antigen present on your red blood cells. Negative means you do not have D antigen. Same rule applies. My body cannot safely receive anything that it does not have. So if I'm type positive, I can get type positive blood because my body knows the antigen. I can also get type negative blood because type negative is the absence of the antigen, and there's nothing there that would set me off. If my blood type is negative, I can only get type negative blood safely, because my body will reject anything type positive because of D antigen. So me, as a B positive person, I can safely get B positive because my body knows both B and positive. I can safely get B negative because I know B and negative is the absence of D antigen. I can safely get O positive because my body knows positive and O means the absence of a letter. I can safely get O negative because my body, there's nothing on it that I can react to. My body will reject anything A or B. If I was B negative, my body would be cool with B negative. It would also be cool with O negative, but B positive and O positive would be bad because I would respond to the presence of D antigen on it. So you can chart this out and you can figure out pretty easily what blood types you can and cannot safely receive. Again, you cannot safely get an antigen your body does not make. So what you're paying attention to is, does my body make A? Does my body make B? Does my body make positive? Because O and negative are the absence of a thing. By the way, what that means, if you're thinking this out, there is a universal donor and there is a universal receiver. Can you think about that for a minute? Universal donor? Hopefully you said O negative, because O negative is the only blood type that lacks antigens altogether. There's no letter group antigen, there's no D antigen. Anybody can safely get O negative blood, so O negative is the universal donor. Blood banks love O negative blood. If you are O negative and you donate a pint of blood, they're going to hound you every single month till you die, begging for your sweet, sweet universal blood that they can give to anyone. By the way, if you're an O negative person, sorry, but the only blood type you can get is O negative because your body will reject every single antigen. So really, frankly, if you're an O negative person, it's in your own best interest to donate a pint of blood once in a while because you might need it. Who's the universal receiver? Think that through. Should be pretty obvious that our universal receiver is AB positive, because AB positive people are the only ones who have all three antigens, A, B, and D antigen. Therefore, their bodies will accept absolutely anything you give them, and all the negative types are fine because that's the absence of an antigen. So AB positive people are the universal receivers. If you're an AB positive person, after you donate a pint of blood and the blood bank sees what type you have, they're not going to chase you down. They're not going to ask you for more, because they don't need to keep a supply of AB positive blood around, because you can literally give an AB positive person any blood you happen to have lying around, and there aren't that many AB positive people. It's the second rarest blood type. Only AB negative is rarer. Yeah, they can give you whatever they happen to have at the time if you're AB positive. They can give you anything negative if you're AB negative. So there's no use in keeping around a supply of special AB positive blood just for you, especially considering that nobody else can get the AB positive blood except for you, because your body is the only one that accepts all three markers. So as an AB positive person, you're the universal receiver. Your blood's not that useful to anybody else. O negative person, you're the universal donor. Everybody wants your blood. Okay, sound good. I've gone slightly over what I like to do with a normal lesson, but you know what, folks? This has been about an hour, but I'm realizing I can actually finish out genetics right here, because we are almost done. I only have one major genetic phenomenon left to tell you about, and maybe a few words about a minor one. So this is the point where I'm going to just carry on this lesson a little bit longer than I normally do. If you feel like pausing it and coming back later, you know, we're about 1:03 right now, 1 hour, 3 minutes, so you know what to do with that. But I'm going to forge on a little bit here. So, anybody know about a condition called hemophilia? Hemophilia. Hemophilia is a rare blood disorder caused by the absence of clotting factors. Your blood is lacking a necessary factor needed to form a clot. As a result, people with hemophilia get injured easily. They bleed profusely, even from small injuries. They heal slowly from injuries. Hemophiliacs can bleed to death even from relatively minor cuts without outside intervention. So a lot of hemophiliacs die young from the various and sundry problems that plague you when you're a little kid and you get hurt. You know about red-green color blindness. If you have red-green color blindness, red and green look the same to you. I'm wearing a red shirt right now, but if you're red-green colorblind, you didn't know that until I told you so, because a green shirt would look exactly the same to you. You have to memorize which one is the green light and which one is the red light in your traffic light, because they're going to look the same when they blink. How about baldness. Any of you guys feel that hairline that's slowly and mercilessly marching backwards and has been doing so since your 20s, and you can't stop it, and you've got that fringe of hair around the back of your head, you know, and just that big bald spot on top? Yeah, male pattern baldness, we call that. Everybody's hair thins out as you get older, but this is a very distinct kind of baldness in which, again, it goes backwards from the forehead, but tends to stick around the edges. What do all three of these conditions have in common? All three of these conditions share in common the fact that men get them way more often than women. And the reason for this is because all of these are what we call sex-linked traits. In sex-linked traits, the gene for the trait is found on the sex chromosomes. That is found in the 23rd chromosome in humans, or whatever chromosome is your sex chromosome in other species. But yeah, it's found on this 23rd chromosome in humans, the one that controls biological sex. Now, you all hopefully remember the 23rd chromosome comes in two varieties, X and Y. If you have two copies of the X chromosome, you will be biologically female. If you have one X chromosome and one Y chromosome, you will be biologically male. The Y chromosome, incidentally, only has 77 active genes on it. It basically contains the genes that are needed to modify base female anatomy into male anatomy. Genetically, we are all female by default. The presence of a Y chromosome flips genetic switches that cause male development. But here's the thing. If you've got a gene on the sex chromosomes, the inheritance pattern of that gene for men and women is going to be slightly different. Let's suppose a gene is found on the Y chromosome, a Y-linked gene, as we call it. Men only have one Y chromosome. It does not do the same things that are on the X chromosome. The genes are not paired off like they are for every other chromosome pair. So if you have only one Y chromosome, you're automatically homozygous for everything on it because there's no possibility of a second Y chromosome there to compete with. Therefore, dominant versus recessive makes absolutely no difference. Every gene on the Y chromosome will be expressed if you have only one Y chromosome. So regardless of whether it would be normally a dominant or a recessive, because it doesn't have any competition, you're homozygous for all of them. So if a condition is Y-linked, if it's a gene found on the Y chromosome, it is always expressed if present, and it's always passed father to son. There's no possibility of a woman getting it because she doesn't have a Y chromosome. Let's, however, suppose that our gene is X-linked. This is actually the fun one. Y-linked is not all that interesting because A, it's pretty rare, and B, because, again, it's either a have it or don't have it. You know, there's no possibility of being a carrier, and it only affects men. But X gets different because everybody's got an X chromosome. Men have one X chromosome, which they got from their mom. Women have two X chromosomes, which they received one from each parent. Now, here's where this gets fun. If you are a woman and you have two X chromosomes, you have the possibility of having a heterozygous situation there, right? You could have a dominant version and a recessive version of genes on the two chromosomes. It matters. So let's suppose a gene is X-linked dominant. It is found on the X chromosome. It is a dominant gene. If you are a woman, you will absolutely express it. Now let's suppose you have some kids. You pass on your X-linked dominant, your X chromosome with the dominant gene on it to one kid, your X chromosome without the dominant gene on it to the other kid. The other kid doesn't have the condition and isn't a carrier of it. Can't be a carrier of a dominant gene anyway. The kid that got it is going to express the condition regardless of whether they are male or female. Everybody gets an X chromosome from mom. So an X-linked dominant condition can be expressed by both men and women. Now let's suppose that a guy has an X-linked dominant condition. When he passes on his chromosomes, each of his kids gets either his X or his Y. If he passes on the Y, the child will be a son. And that child will not have that condition because it's an X-linked condition. You can't get it from dad because dad gives you a Y. That's why you're a son. But dad can't pass that condition to his daughters. So a dad with an X-linked dominant gene will give it to all of his daughters. His daughters are guaranteed to have it. None of his sons will have it guaranteed unless they got it from mom. So that's an interesting thing with X-linked dominant, that the inheritance pattern is that both men and women are equally affected by it, But men can only pass it to their daughters, not their sons. Women can pass it to all of their kids. The interesting one, the really interesting one, is X-linked recessive. When we talk about sex-linked traits, we are almost always talking about X-linked recessive traits. In fact, all three of those diseases I mentioned, male pattern baldness, red-green color blindness, hemophilia, and a couple other ones, muscular dystrophy is an example, all of those are examples of X-linked recessive conditions. Now, this is why it's interesting. You're a woman, and you inherit an X-linked recessive gene for hemophilia, let's just say, from one of your parents. But you get a good X from the other parent. Good X is dominant. Bad X, let's call it, with the hemophilia gene on it, is recessive. You're heterozygous. You are a carrier of hemophilia. You will not have it because the dominant gene wins when you're heterozygous. And hemophilia is a recessive condition. So a woman with one hemophilia X and one normal blood X is not going to have hemophilia. Now let's suppose that woman has some kids. Right here. This is what happens if that woman has some kids. You see that she's got one X chromosome. We put the little superscripted H next to it to indicate that that's got the recessive gene for hemophilia on it. She's got one normal X chromosome. She's got a 1 in 4 chance of having a daughter that is a carrier like her. She got bad X from mom, good X from dad. This couple has a 1 in 4 chance of having a daughter that is completely unaffected. She does not have the gene for hemophilia at all. She's not even a carrier. She can't pass it on. This couple has a 1 in 4 chance of having a completely unaffected son. Got good X from mom, got dad's Y. But check it out. This couple has a 1 in 4 chance of having a son who got bad X from mom and Y from dad. Remember how I said men are homozygous for everything on the Y chromosome because they only have one? Men are also homozygous for everything on the X chromosome because they only have one. There's no possibility of having a dominant gene on the other X to compete with it because men don't have another X. The X and Y don't contain the same genes. Therefore, men are going to be homozygous for everything on the X chromosome, regardless of whether it is dominant or recessive, and they are therefore going to express that gene. So in other words, a man only has to inherit one copy of an X-linked recessive gene in order to suffer from it, because he only has one X chromosome. He only has to get it once. That is why X-linked recessive conditions are way more common in men than they are in women. The incidence of hemophilia in men is about 1 of every 5,000 men has hemophilia. Only about 1 of every 30,000 women has hemophilia. The inheritance pattern, therefore, is mom to son. You expect X-linked recessive conditions to go from mom, who is a carrier, to son, who is afflicted. There is also a pattern from father to daughter, because you have dad is going to pass on his bad X to every single daughter he has. But she'll be a carrier unless mom also gives them a bad X. Dad can't pass it to his sons. If you've got a hemophiliac father and you're a biological male, you didn't get hemophilia from him if you have it. You couldn't because he gave you a Y chromosome. That's why you're a guy. And as a consequence of that, men with X-linked recessive conditions cannot pass them to their sons because sons get the Y, but all their daughters are going to be carriers. And so guys, if you have that receding hairline and you're not happy about it, blame mom. It's not dad's fault, even if he had the same thing as you. All right, can a woman have this? Let's check it out. Got some other possibilities here. Here is dad. Here's dad from the previous generation. This is the F1 generation. We got a guy with hemophilia. Now, hemophilia guy here partners up with an unaffected woman. Here are the way the kids come out. They've got a 1 in 4 chance of having, actually they've got a 2 in 4 chance of having carrier daughters. Every single daughter gets dad's bad X. Every single daughter is getting a good X from mom. So all of them are going to have one copy of the good dominant gene and one copy of the recessive gene for hemophilia. These girls will all be carriers of hemophilia. This couple will always have unaffected sons. because dad is giving his sons his Y chromosome, and even though he has hemophilia, the gene for hemophilia is not on the Y, it's on the X. They're getting a good X from mom, they're getting a good Y from dad, unaffected sons, carrier daughters. Here is how a woman can have hemophilia. Here is affected dad and carrier mom. Look at this couple. They have a 1 in 4 chance of having an affected son. He got bad X from mom, he got Y from dad. So even though dad was hemophiliac, this guy got it from his mom. Just keep that in mind here. That's an interesting thing, right? You know, dad is hemophiliac, but he can't have a hemophiliac son. So if he does have a hemophiliac son, the gene came from mom. This couple has a 1 in 4 chance of having an unaffected son. Got Y from dad, got mom's good X. This couple has a 1 in 4 chance of having an unaffected daughter. She got good X from mom, bad X from dad. She's a carrier of hemophilia, but she herself does not have it. She can have kids who do, but she doesn't. And this couple has a 1 in 4 chance of a hemophiliac daughter. Because both mom and dad have one copy of bad X, there's a 1 in 4 chance that they'll have a daughter that got both of those. And that is the only way that a woman ends up expressing an X-linked recessive condition. If dad has it, and mom either has it or is a carrier of it. I think you can see now why X-linked recessive conditions are way less common in women than they are in men. 1 in 10 men, approximately, is red-green colorblind. Only about 1 in every 1,000 women is red-green colorblind. Similar pattern holds up with muscular dystrophy. It's a much rarer condition in women than it is in men. Male pattern baldness is the most common of these, which should be unsurprising because it's the least lethal of them. It's the one least likely to cause you problems that are going to cause issues in your life. And there, as concomitantly, there are more women affected by male pattern baldness than there are women affected by hemophilia or by red-green color blindness, but it's still not common in women, right? We call it male pattern baldness for a reason. So, it is technically possible for a woman to have an X-linked recessive disorder, but she has to inherit it from both parents for that to be the case. More often than not, it's going to be men with one copy of the X, got it from their carrier mom, and then those guys pass it on to daughters who become carriers themselves. All right, so that is sex linking. One last genetic phenomenon I want to tell you about, and it has to do with a different portion of your DNA. Actually, we're going to talk about 2 last genetic phenomena. One, I want to talk about very briefly a thing called epigenetics. We're not going to go into this in much detail. If you take a genetics class, which you should, then we'll talk about it more there. But epigenetics refers to the things that affect the expression of your genes beyond the genes themselves. Basically, molecules that can attach to the DNA that can turn a gene on or turn it off or cause it to be expressed more or expressed less. There are a lot of environmental factors involved in epigenetics that switch genes on or off at different points in your lifetime. Certain points in your life cycle, some genes are active that are not active in others. You've got genes that are active during your growing years that are not active as an adult. There's a whole suite of genes that turns on at puberty. A lot of things like that. There are also environmental factors that can affect genes, causing them to switch on and switch off. We have learned, for example, and here's a really interesting phenomenon, that some of the genetic switches that are flipped in people who end up with PTSD, some of the trauma from PTSD, actually can cause tags of sorts to be attached to the DNA, which means that some amount of trauma can actually be inherited. That once that tag is attached to your DNA, you can make a gamete that has that tag on it, and it's conceivable that your kids could inherit some genetic problems that come from your preceding trauma. There was a really interesting study done of the descendants of Holocaust survivors that find that certain disorders certain psychological disorders are more common in Holocaust survivors than in the population at large. That it seems like there was literally some inherited genetic aspect to the trauma, that their DNA was modified by an epigenetic tag created by the chemical response to the brain chemistry as a result of trauma that led to the PTSD. I can't tell you a lot about epigenetics here. It's a really complex area, but suffice to say, not every gene in your life is active at all times, and although the environment does not cause you to gain new specific genes, it can modify the expression of genes in some really interesting ways. And the last phenomenon I wanted to tell you about has to do with something completely different. Now, I've just told you about a whole lot of different genetic conditions. Autosomal DNA, you might have heard that term autosomal, A-U-T-O-S-O-M-A-L. Autosomal genes are those found on chromosomes 1 through 22. You might say quote-unquote normal genes on your regular chromosomes. Sex-linked genes are found on chromosome 23. But there's another category. The other category is mitochondrial genes. Remember how your mitochondria has its own DNA, non-chromosomal DNA, inherited from the bacterial ancestors of all mitochondria. And whenever your cells divide, they send a signal to the mitochondria saying, hey, time for that division thing, and the mitochondria also divide, copying their own DNA dutifully out. Mitochondrial DNA basically controls mitochondrial things. A couple of other functions it has in the cell because it's producing protein products that can affect the cell. We have learned that a couple of genetic disorders are actually, in point of fact, linked to your mitochondrial genome. There's some evidence to suggest that diabetes, for example, can be linked to mitochondrial DNA. But anyhow, here's the interesting thing about mitochondrial disorders. Where'd you get your mitochondria from? Think back. From mom. Your mitochondrial DNA in almost all instances is identical to that of your mother because the egg cell contains mitochondria, but the sperm, the part of the sperm that penetrates the egg, does not contain the mitochondria of the sperm. Therefore, except in very, very, very rare instances, dad's mitochondria do not become part of the zygote when the egg is fertilized. And therefore, except for those very, very, very rare instances, which we only discovered that was even a possibility last year, all your mitochondrial DNA came from your mother. Now, mitochondrial DNA is non-chromosomal. In other words, no paired genes. Therefore, dominant versus recessive means nothing when it comes to mitochondria. You're homozygous for everything in the mitochondrial DNA. It's either there and you express it, or it's not there and you don't express it. Now, if mom has a mitochondrial disorder, all of her kids will receive it because all of her kids get their mitochondria from mom's egg cell. Again, dominant versus recessive doesn't matter. The gene's either there or it's not there. And if it's in mom's mitochondria, it will be in every egg she makes. Ergo, if mom has a mitochondrial disorder, every gene in her mitochondria ends up in all of her kids. 100% transmission. Even if one of dad's mitochondria got in there somehow, mom's are still going to be there. If a guy has a mitochondrial condition, he never passes it to his children because his mitochondria do not become part of the zygote. Again, there's that very, 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 very rare instance we have discovered recently in which there's some small amount of mitochondrial transmission from the father, but it's like 1 in every 200 million, so basically never. But under normal circumstances, yeah, you didn't get any mitochondria from your dad. So if you have a mitochondrial disorder and you're male, you will never pass it on to your kids. So the inheritance pattern with mitochondrial disorders tends to be women always pass them on to all of their children, men never pass them on to any of their children, and if they are present, they are always expressed. Dominant versus recessive is irrelevant. Okay? I guess to tie things off, let's just say a few words about this thing we call a genetic disorder in the first place. What is a genetic disorder? We mentioned this a little bit in our chapter on mutation. A genetic disorder basically is the result of a negative mutation, that it is a negative allele that came about, that a mutation modified an existing gene in a way that made it work more poorly. It made it do something wrong that it should not be doing. Ergo, your body cannot form one or more proteins properly, and you have problems as a result of that. Now, here's the thing. That mutation only had to happen once. If you're born with that mutated gene, assuming it doesn't kill you, and you live to be old enough to reproduce, you can pass on that gene to your offspring. In other words, that mutation just had to happen once, and now that gene, that new allele, is a new variation present in the gene pool in the population that can be passed on like any other variant. And as long as you have kids, it's going to persist. So just because you have sickle cell anemia does not mean that a novel mutation happened in you or in your parent to give you it. Odds are pretty good that you just inherited the gene from somebody else who inherited it from somebody else who inherited it from somebody else, probably going back hundreds of generations or thousands of generations. We define a genetic disorder, of course, as being a gene that causes problems for you. It's a situation in which the gene makes life difficult for you in some fashion. Genetic disorders tend to be rare in direct proportionality to how lethal they are. If a genetic disorder always kills you as a kid, it's going to be very rare. Tay-Sachs syndrome is very rare because nobody survives past the age of 5 with it, and therefore everybody born with Tay-Sachs syndrome dies before they reach reproductive age. By contrast, say you've got hay fever, probably not going to die from it. It is technically possible that you could have such a severe response that you die of anaphylaxis, or that bunged up stuffy nose made one of your ancestors not be able to breathe as well and couldn't outrun a saber-toothed tiger or whatever. So technically, there might be some people who die of hay fever, but not very often. So that one tends to stick around in the population because odds are good. It is not reducing your likelihood of successfully reproducing. Ergo, that gene is going to stick around. Something that's more middle ground, like asthma, for example. I'm asthmatic. Not everybody's got asthma, but asthma is a condition you're likely to survive into adulthood with, and therefore, especially with modern medical intervention. Ergo, if you're an asthmatic, you're probably going to get reproductive opportunities. Yeah, there are going to be people who die young of asthma. There are going to be people who die of complications of asthma, like pneumonia or whatever that we asthmatics are prone to. Those people might get taken out of the gene pool before they get a chance to reproduce. But odds are it's probably not going to happen. So asthma sticks around. But because it does kill some of the people with it, it's a bit rarer than, say, hay fever. You might be asking yourself, how the hell does Tay-Sachs stick around if it is lethal to everyone who gets it? Because it's recessive. Do not fall into the trap of thinking recessive equals bad, but there is a reason why a lot of genetic disorders are recessive, for the very simple reason that, by definition, a genetic disorder causes problems for the organism that has it. Ergo, it reduces their likelihood of successfully reproducing, even if only by a little bit. Averaged out over the population, that means that gene becomes less common over time. So what this means is if my genetic disorder is recessive, it's going to tend to take people out of the gene pool and therefore become less and less common across generations. But because it is a recessive disorder, that means that it can be kept around by plenty of people who aren't suffering from it, and therefore it's not causing natural selection to act against them. So if everybody who is born with Tay-Sachs syndrome dies from it, that's unfortunate. Unfortunately, because it's a recessive disorder, that means that there's kids with Tay-Sachs syndrome probably have siblings who have one copy of the gene, who are carriers of the disorder. Therefore, they are not affected by it, and they can pass it on to their kids. That's the only way a guaranteed lethal recessive like that stays in the population, but you can see why it's vanishingly rare. So it's not so much that recessive makes you more likely to be a disorder. It's more that a disorder is more likely to persist if it is recessive. Dominant disorders have a harder time sticking around. They do happen. Huntington's disease, terrible genetic disorder that's pretty much guaranteed to kill you if something else doesn't kill you first. It's a dominant disorder. How the hell is that one stuck around? Simple. It doesn't affect you till middle age or later. Therefore, most of the people who have Huntington's disease have already reproduced by the time they find out they have it. And therefore, the gene is stuck around in the population. But because it's automatically lethal, it's relatively rare, because some of those folks do get taken out before they get a reproductive opportunity. So that's really all that you can say about genetic disorders. Some are dominant, some are recessive. They can follow any of the disorder patterns I've talked about. We've learned that a lot of them are polygenic. Some of them are pleiotropy, as you saw with sickle cell and achondroplasia. It's hard to make generalizations about them. But I will say this about them. The environment you live in also might determine whether your condition is a disorder or not. If you live in Western Africa, you might be real happy to have one copy of the sickle cell gene because it makes you malaria resistant. Here in the United States, where there is no malaria, all that means for you is that if you happen to find yourself in Western Africa, you don't have much to worry about. But in the meantime, here in the US, it means you have a chance of having kids with sickle cell anemia. So we tend to think of it as exclusively a bad thing here. Let's suppose you're a little white bunny rabbit hopping around in the park in a temperate environment. Little white bunny rabbit gets spotted by foxes and cats really easily, can't hide as well. Little white bunny probably dies before it gets to reproductive age. Put that white bunny in Alaska. Arctic environment, white bunny blends in real nicely. Blends in better than brown bunny. Ergo, white bunny has an advantage. So sometimes whether or not a condition is a disorder really depends on the environment that you see it being expressed in. What can save you in one environment might kill you in another. But I am at this point straying into content relevant to my Bio 2 class, so maybe I should call this a night. And that, folks, wraps up our chapter on genetics. I probably could have gone into a second lecture, I'm realizing at this point, but, well, fourth lecture. But you know what? It's over with now. So I hope you all enjoyed this series, and I hope you all have a very good night. I'll talk to you soon.